Ever imagined your cells not as static blobs, but like adventurers?
Yeah.
Responding to signals, navigating chemical landscapes.
Yeah, it's wild. Really dynamic, especially when you look at ion carriers shaping their movement and internal environment.
Oh, totally. I think most people picture cells moving with like those tiny feet pushing them along, that actin stuff. But today, we're going deeper, like how these ion carriers make gradients inside that do way more than basic movement.
It's like this.
Mhm.
Even when a cell's not moving, there's a ton going on. Ion carriers are constantly working right, keeping ions balanced, pH levels right, electrical charge steady inside. It's not all the same in there. The cell uses these subtle gradients to its advantage.
So even when a cell's just hanging out, there's a whole hidden world.
Absolutely. These gradients are key to how cells react to changes around them and even move in certain directions.
Okay, let's get into these gradients. We know cells can be polarized, but what's that really mean here?
When we say polarized, think about an uneven spread of those ion carriers on the cell membrane. It's like different neighborhoods in the cell, each with its own special ion carrier population.
So it's not just having them, it's where they are on the cell that matters.
Precisely. Like the sodium-hydrogen exchanger, or NHE. Crucial for cells migrating, think cancer cells spreading, that kind of thing. More NHE on one side can actually pull water in, making a flow that pushes the cell forward.
So a cell can make its own currents to move itself. That's
Wow.
crazy. What are some other players on this ion carrier team?
Well, besides NHE, which does both pH and electrical stuff, there's AE2. That's a chloride bicarbonate exchanger, kind of the master of pH inside, making sure it's not too acidic or alkaline.
Keeping that pH balance seems super important.
Absolutely. Then you got passive potassium and chloride channels. They mainly regulate the electrical potential, across the cell membrane, like a tiny battery powering different processes.
So we have this whole system of different ion carriers, each with its own job, like a finely tuned orchestra keeping things harmonious inside.
Perfect analogy. And it's not just internal harmony, it's about responding to what's going on outside, too. Imagine the cell hits a gradient of pH or electrical potential outside. It has to adapt to keep its own internal balance.
So how does it handle those outside changes without getting totally thrown off?
What's fascinating is it acts like a buffer. Say there's higher pH outside. The inside pH will change a bit too, but not as much. It's like the cell saying, hey, I see that change out there, but I'm going to minimize its effect in here.
That's smart. How does it even sense those outside changes, though? Does it have some kind of sensory system?
It does. Specific ion carriers are the sensors. Like A2 is super sensitive to pH changes, while passive potassium channels are tuned to shifts in electrical potential. They send info about the outside back to the cell so it can adjust.
So the cell is always checking its surroundings, making tiny changes to stay balanced.
Exactly. This constant fine-tuning is key for survival and for the cell to do its job.
This is way more dynamic than I ever thought. Gradients, specialized ion carriers, back and forth with the environment. It's like a microscopic dance.
It really is. And we're just starting to understand how this affects everything from movement and communication to growth and development. But to get how these gradients turn into action, we got to talk about the cell structure, particularly something called F-actin.
Okay, I'm intrigued. Tell me more about this F-actin and how it fits into everything.
Think of F-actin like the scaffolding of the cell, Keeps its shape, provides support. But here's the thing. It's not just static. It's super sensitive to pH, believe it or not.
Right. The cell's support structure changes with pH. How does that even work?
Basically, pH changes can mess with how fast F-actin puts itself together and takes itself apart. So even a small pH shift inside the cell can make that whole scaffolding rearrange.
Well, so it's not just like little ion changes. The whole cell structure is affected by these gradients.
Exactly. And remember how we talked about the cell being a buffer for pH changes outside? Well, that buffering creates a pH gradient inside too, just smaller. And that gradient can cause F-actin to assemble and disassemble differently in different parts of the cell.
So like one side might be slightly more acidic, leading to different F-actin structure.
Yeah.
But how does that actually make the cell move?
Picture this. On the side with the higher pH, F-actin might break down faster, becoming more fluid, less dense. Opposite side, lower pH, F-actin's packed tighter. That difference in structural strength can make the cell lean or even push itself in a certain direction.
So the cell is kind of pulled towards the more fluid side, guided by this pH-sensitive scaffolding.
Yeah, you got it. That's one way gradients, in this case pH, can cause movement. The cell can use similar tricks, playing with different gradients and ion carriers to navigate its surroundings. But get this, there's an even crazier connection between F-actin and ion carriers. We know F-actin changes with pH, But the F-actin itself can actually affect where those NHEs, remember those sodium-hydrogen exchangers, end up.
Hold on. The scaffolding tells the ion carriers where to go. Isn't that backwards? How's that even possible?
It's this protein called ezrin. It links F-actin and NHE, creating a feedback loop. The cell can basically control its internal water flow by messing with where the NHEs are through the scaffolding, like building dams and canals inside to direct the water.
So the cell isn't just reacting to gradients. It's actively shaping them using its own structure. That's wild.
It shows just how complex and adaptable cells are, and this whole water flow control is especially important when there aren't many cues from the outside. Even without external gradients, the cell can still make its own currents to move and keep its internal balance. Now, we've mostly talked about how cells react to gradients that are already there, but what if they could make their own?
You're saying cells might generate their own gradients to talk to each other, like some kind of secret network?
That's one of the big questions researchers are looking at. It's like cells whispering to their neighbors, coordinating their moves and responses.
That's mind-blowing. And it's all thanks to these tiny ion carriers. They're like the unsung heroes of the cell world.
For sure. And remember, this isn't just about movement, it's also about keeping that stable internal environment we talked about. Think of it. The cell membrane is like a gatekeeper, controlling what comes in and out. Ion carriers are like the guards on that membrane.
So they decide which molecules get through. How do they know which ones to let in and keep out?
It's all about those gradients again. Cells want a specific environment inside with just the right balance of ions, pH, and all that. So they use the fact that molecules naturally move from high concentration to low concentration.
So if there's more of something outside, it wants to flow in to even things out.
Exactly. And the cell can use that. It puts channels in the membrane, letting certain molecules flow down their concentration gradient, like opening a gate and letting them rush in.
But what if the cell needs to move stuff against that concentration gradient?
That's where pumps come in. Cells have special pumps, like the sodium-potassium pump, that can move ions from low to high concentration, but it takes energy to do it.
So it's spending energy just to keep things balanced, like a constant juggling act.
You got it. But it's super important for the cell to survive and do its job. It's dynamic, with channels letting stuff flow down gradients and pumps working against them to keep that balance. So we know ion carriers make gradients and maintain balance. We know gradients can make the cell move. But can different types of gradients, like pH and electrical potential, work together?
That's a good question.
And the answer is absolutely yes. They actually work together to create some really incredible things.
Like what? Give me an example.
Let's say there's a higher pH outside the cell, meaning it's more acidic inside. Well, that pH gradient can actually affect how other ions move, like the ones involved in electrical potential. It's like a domino effect.
So one gradient can trigger changes in another. It's all connected.
Exactly. The pH gradient might make certain ion channels open or close, changing the flow of ions that control electrical potential. It could even affect how those ion pumps we talked about work. It's this complex web of gradients all working together that allows the cell to respond to its environment in all sorts of ways.
Okay, so we've got this whole system of ion carriers making and controlling these gradients. And we've got these different types of gradients interacting with each other. But how does the cell actually use all of this to, like, move in a specific direction? We know it can create flow, but how does it steer?
It all comes back to that structure we were talking about, the F-actin, how it's sensitive to pH.
Oh, right. It can, like, reshape itself based on the pH changes.
Yeah. So imagine, like, the cell senses a higher pH on one side of its environment. That kicks those ion carriers into gear, making an internal pH gradient that kind of mirrors what's going on outside.
So one side of the cell basically becomes more acidic than the other.
Exactly. And that internal pH gradient then affects how that F-actin puts itself together and breaks apart. Remember, F-actin is like the scaffolding of the cell, giving it shape and support.
So we're saying the cell scaffolding is being, like, remodeled based on this pH gradient. How does that actually make it move in a specific direction, though?
Okay, picture this. On the side where the pH is higher, that F-actin breaks down faster, so it becomes less dense, kind of more fluid. But on the other side, where the pH is lower, the F-actin is packed together tighter. This difference in, like, how strong each side is creates a kind of imbalance, and the cell sort of leans towards the more fluid side.
So it's almost like the cell is being pulled in a specific direction by how its scaffolding is changing.
That's a great way to put it. This is just one way these gradients can be used to make directed movement. The cell can use similar mechanisms with different gradients and ion carriers to find its way around, in its environment. It's amazing how much is going on at such a tiny scale.
It really is. Makes you think about life in a whole new way. We've covered so much today, from how actin makes cells move to this whole intricate dance of ion carriers and gradients. We've seen how cells keep things balanced inside while dealing with a constantly changing world outside. And we've even gotten a glimpse of how structure and function work together to let cells move in such a dynamic way.
It's been a really cool journey into the microscopic world.
And to think we've only just scratched the surface. Who knows what else we'll find as we keep exploring the cell?
There's definitely a lot more to uncover about how they sense, respond to, and even control their environment. It's a never-ending journey of discovery.
Well, I'm definitely excited to see where it leads. Thanks for joining us on this deep dive into the amazing world of cells.